Welcome and thank you all for attending the second session of GenScript ProBio's Innovative Antibody Drug Discovery Research Webinar Series. Today I'm going to talk to you about single-cell technology and its application for antibody drug discovery. The webinar is going to be divided into 4 parts: the significance, the approaches, single-cell technology for antibody drug discovery, and ADD platform comparison. So why do we need to access cellular information at the single cell level rather than looking at the average characterization over a cell population? The short answer is that no two cells are alike. Recent advances in cellular profiling have demonstrated substantial heterogeneity in cells that are once deemed identical. Heterogeneity is a critical aspect in understanding cell biology, especially in immunology, oncology, or cell differentiation. To give you a few examples, we can see that the expression of a specific gene or protein is vastly different if you look at different cell types or between progenitor cells to their differentiated cells. The generation of enormous B-cell repertoire from B-cell recombination event essentially takes place on a one-cell, one-antibody basis, where a single mature B-cell produces a single antibody. The cellular response to any external stimulus, for example in the case of drug resistance developments, varies from cell to cell as well. Here, if you look at the cellular response measured by the expression of a set of genes, or the covariance of the expression of different genes is in fact bimodal, where there are 2 peaks, that means we will get completely the wrong idea to look at the average behavior of a cell pool with some averaged value of the bimodal peaks. The nature of why we need a single cell level resolution or to analyze individual cells also require the scale of single cell analysis to be high enough to overcome variations that arise from technical noise and intrinsic cellular differences. Meaning that if the reason why we need to look at single cells is that each cells are different, then we need to make sure we look at enough number of single cells to increase the confidence in our data and ensure any interpretation is statistically meaningful. Here we can see that the scale of single cell studies, or the number of single cells per study, has exploded from a single digit to the millions in the past decade or so with the technical advances in microsystems. And those systems typically will focus on improvements in elements such as throughput, information content, biological context, and the ability to perform manipulation to characterize or understand the cell of interest. There are many inherent advantages of these physically compact microsystems, such as that they are generally require a much smaller amount of reagents that are used, which in turn brings down the cost, and by decreasing the scale or the volume of experimental system, you increase the concentration or the content or the subject of your analysis, which essentially means that you can increase your assay efficiency. So after we talked about the significance of single-cell technology, and now we're going to move on to different approaches of single-cell technology. So here you can see a few examples of different types of microfluidic tools that are commonly used for single-cell analysis. First is what we call a valve-based tool. Basically, you have a microfluidic system to flow in cells into a channel with built-in valves that can be pressed down or opened by pressure and expand perpendicularly to the flow channel and trap a cell in between valves. And obviously, the number of single cells that you can examine is completely dependent on the number of channels or valves that you have built. And these type of valve structures are typically prone to biofouling, which is the accumulation of cell debris or biological substances on the surface of these valves, which limit the number of repeated use. And the structural constraints and biofouling issue have greatly limited the market success or the market use of these type of microfluidic tool. Next, we have a droplet-based technology. Here, the schematic shows a cell suspension passing through a channel, and you deliver a carrier oil to capture the cell in suspension for single-cell separation. A different case of droplet-based microfluidic tools is fluorescence-activated cell sorting, which does not fall into the typical definition of droplet, where there is no emulsion or oil involved, but it does rely on breaking a stream of liquid or the cell suspension into individual droplets as well. We'll talk a little bit more about these two types of microfluidic tools in the next few slides, given their popularity. And lastly, we have a nanowell-based technology where you can see that each well or each cell is confined at a fixed spatial location or in the nanowells. And the cells can be loaded by gravity or more sophisticated method. This is useful for conducting multiple measurements, but the throughput is somewhat limited to the number of wells or nanowells that's fabricated for your system as well. Here's a more detailed illustration of emulsion droplet technology for single-cell sequencing. And what you can see is that the cell and lysis buffer together with RNA capture beads are fed into the nozzle connected to an oil pump to deliver the oil to encapsulate single cells. And once the emulsion is formed, the droplet will be broken off, ideally containing one emulsion particle per droplet, where the cell lysis and RNA capture can take place. Then the droplet will be collected and purified for downstream analysis. This emulsion droplet system has very good throughput and very high speed. But you can imagine it is very difficult to gain specific control to any one particular droplet, and it requires a significant amount of optimization, such as the flow rate of the liquid phase and then the oil, and the uniformity of droplet breakage for each droplet to be precisely the same size with the same amount of content. For example, here cell lysis buffer or your RNA capture beads. And typically the capture rate is not 100% successful, and it can be biased towards capturing larger cells. Another very popular and well-established single-cell microfluidic system is FACS. First, the cells will be labeled with fluorescent antibody molecule against a specific or more than one specific cell surface proteins. Then the labeled cells in suspension will be entrained in the center of the narrow and rapidly flowing stream of liquid. The liquid stream is then broken into droplets by vibration, and based on the light scattering generated due to the fluorescent characteristics of each cell, the droplets will be charged and eventually diverted into different collection centers with an electrostatic deflection system. FACS can also work extremely fast and with very high throughput, and allows for a multi-parameter detection. The sensitivity can be somewhat compromised, and depending on how you label the cells, and then typically the higher the throughput, the lower the sensitivity of your system. The cell labeling inherently limit the use of FACS to only work for cells with known surface marker. For example, a membrane-bound BCR, and the example that I'm showing you right here is where you stained the antigen or the BCR on the memory B cell and using the FACS for sorting those type of cells. But if your cell of interest do not have a known or specific cell surface marker, then you can't really use FACS for those type of cells. And typically it lacks the functional analysis as well, and the cell viability can be compromised for the labeling and the sorting process as well. So far, we've went over some of the platforms for single-cell technology and of how they work, their advantages and disadvantages. But here I want to summarize for you the desirable features that are needed for single-cell technology-based antibody drug discovery. And those include throughput efficiency, because there's an enormous amount of immune cells to assess. the sensitivity, the ability to perform multiple analysis and not just limited to antigen specificity, but also include a functional aspect. And finally, we want a system to work directly with antibody secreting plasma cells that do not have a membrane-bound BCR. And with that in mind, I want to introduce you to our newest addition in antibody drug discovery at GenScript ProBio, the single B-cell antibody screening platform. First, you can see an overview of our ProSpeed platform with the processes and timeline. We start off with immunization with standard immunization, which is about 8 to 10 weeks, and express immunization, which is about 2 weeks. Then the spleens, bone marrows, or lymph nodes, depending on the immunization method, will be harvested for CD138 enrichment. And after that, the enriched plasma cells will be imported or to be penned, and you will see why it's called penning in the next few slides. And after the single cells are loaded, we can perform various assays, including testing for specificity, affinity, and functionality. And after the clones of interest are analyzed and identified, B cells will be exported into lysis buffer for downstream sequencing and recombinant MAB production. Here we want to highlight that from harvesting the B cells to lead identification, it takes less than 1 day, which is dramatically shortened compared to typical hybridoma method. And from the beginning of immunization to small-scale MAB production for in vitro validation, this total duration is 3 to 3.5 months if we went with standard immunization, and only 1 to 1.5 months with express immunization. And with the next few slides, I will talk to you more in details about how this highly automated, efficient, and comprehensive system works and why it can work so fast to enable this tremendously expedited antibody drug discovery process. There are three main steps that make up the general workflow from the single B-cell screening using the beacon or the ProSpeed system. First is importing the cells. Next is assay, and finally exporting the cells. So the importing step, or what we call penning, involves light activated identification of each single cell and caging them. So I'm going to play the video for you where you can see that all those little cages are actually capturing each individual cells, and it's bringing the cages down to what we call the pen. So that this is why the whole process is called penning as well. Okay, so here you have the cells that are being penned into the nanopens. So next, after the cells are imported or penned, we start the assay. So I'm gonna go back and play the video for you as well, so you know what I'm talking about. Okay, so for the assay, I'm showing you the video right here is where the secreted antibody from each pen are being captured at the opening of each pen using antigen conjugated beads. And you can see that the signal is actually being recorded over time, so it's not just it's not just an endpoint measurement, but a time resolved measurement. Therefore, and then what you can see is that as the antibody is being secreted and accumulated at the opening of each pen, you can observe what we call the fluorescent bloom. And because of the way the assay results is visualized or recorded, this is highly reliable. compared to the endpoint measurement, and multiple assays can be conducted either sequentially or simultaneously as well, depending on how you design your assays. So essentially what we want is those clones that have the fluorescent bloom at each opening of those nanopens. Okay, so after we done the assays and identify our cell of interest, we'll start exporting the cells. So what you can see right here is that the OEP, or the what we call the optoelectropositioning system, is moving this one particular plasma cell out of the pen. And then the cell will be exported all the way to the channel and flushed into 96 well plates pre-filled with lysis buffer. Here you can see that the plasma cell is actually being moved to the channel. And then the plate or the cell in lysis buffer can be stored at -80 degrees before sequencing. Okay. So now you have seen how the system works. You might have some idea of why the single B cell screening is so much faster or more robust with just one cell. So I'll walk you through a simple math to put it kind of all together. For single B cell screening, the media volume per pen is about 1 nanoliter, where each pen has one cell, so the concentration is 1 cell per 1 nanoliter. Although the effective concentration, and basically what I did here is to convert the unit to the number of cells per ml, then you get 10 to the 6 cells per ml. Now, if we look at hybridoma or ELISA, the media volume is 100 microliter, and typically you can get an ELISA read with about 10 to the 4 cells, or that's the number of cells that you have to grow up for an ELISA test. So here your concentration is 10 to the 4 cells per 100 microliter, or 10 to the 5th cells per ml. So the effective concentration is actually 10 times higher with single B cell screening, which means higher robustness and higher sensitivity. And since you don't have to wait for the cells to grow up to a certain density to assay, the assay speed is within minutes rather than days. Here I'm going to present you a case study to illustrate the workflow establishment from immunization to sequencing. With a little over 5,700 cells, we generated 7 leads, which the affinity or antigen-specific binding for all 7 were validated to be at the nanomolar range. Not only we can screen for antigen-specific binding, we can also have a selection of different assays according to the clients or according to your specific need for each specific project. And those include cell-based bindings, or screening for antibody that will bind to a target antigen expressed on a stable cell line, or bindings identified through target antigen conjugated to beads, cross-species reactivity, or antibody that binds to a different epitope than a positive control antibody if that is available. Despite of the different types of assay, this process is still completed within less than 1 day. The multiple assay types is not only useful to scale up the information content we can collect about each lead, but it's also critically important to narrow down the desirable leads and identify a much more manageable lead number for sequencing and MAB production. For example, in this particular case, we got 169 cell binders, which will be a pretty daunting number of leads for sequencing and MAB production. But with the second assay, we were able to identify ligand blocking activities, and now we narrowed down the number of desirable leads from 169 to 68. And with different types of immunization method or antigen type, the positive rate for cell binders ranges from 0.07% to 1.2%. With bead-based screening, the number is generally higher, from 24 to 474 binders, or the positive leads. So this is not positive rate, but the actual number of binders. I would like to also note that we did a direct comparison of standard and express immunization with the same target antigen. So what we can see here is that with express immunization, we got fewer leads, but it still was able to deliver 48 leads, and it saves you for additional six weeks. And to summarize, with the opto-electropositioning system of this platform, we can precisely control the movement of single cells. The entire workflow is fully automated from import, screening, or assay to export to increase the overall efficiency of screening by shortening the screening process from 3 months in hybridoma to 1 day. The resolution or the level of access to the vast antibody repertoire is at least 100-fold higher compared to hybridoma as well, without the B cell loss or at the fusion step. The platform is also capable of in-depth characterization with versatile and flexible assays that's tailored to any specific projects or any specific needs. So far, I've talked to you about the impressive features of single B-cell screening, but at GenScript ProBio, we also provide screening technologies of different platforms as well. So in a snapshot, we have 3 main platforms, namely hybridoma, which can be coupled with regular transgenic mice, library screening including human naive and synthetic library, and llama naive library, and finally the single B-cell screening featuring the beacon system from Berkeley Lights. So what you can see here is that each platforms have its unique advantages and also some disadvantages. For example, the hybridoma technology is extremely well established, with accumulating over a decade of expert experience, but it is rather laborious and time-consuming. Whereas the single B-cell platform is an emerging new technology, but now it enables the turnaround time to be shortened to as little as 1 to 1.5 months. And of course, with library, one of the unbeatable features is that it does not require immunization, and it's ready to screen with any available antigen. With that, so what you're looking at here is that each platform is not really competitive. It doesn't have to be competitive. I don't have to beat the hybridoma platform with the single B-cell platform because it's so much faster, but it's rather complementary. So the pros of hybridoma is complementary to the cons of single B, or the pros of human naive library is complementary to the cons of single B-cell platform as well, which is where the arrows are pointing. Sorry that it came out a little late. With that, I want to summarize this webinar and leave you with some take-home messages. First, I talked to you about how experimental platform at single-cell scale is vital and transformative. And microfluidic tools have really boosted the development of single-cell technology. Aside from single-cell resolution, antibody drug discovery might require some unique features that's fulfilled by GenScript's ProSpeed platform. And finally, all antibody discovery platforms can be seen as complementary rather than competitive. Thank you for all your attention, and now we're gonna take some questions. So the first question, if the immune response is good, can we say the success rate is almost 100%? Well, I think it depends on how we define successful. If we are looking for a certain number of binders or leads, and that's very difficult to guarantee with any discovery platform. But if we define successful as obtaining any binder, then especially with protein-based binder, the success rate is almost 100%. How reliable is the in-chip assay, or do we do the in vitro validation after the screening on chip? It's close to 100% so far, although we've only completed in vitro validation with 2 projects. We got 100% leads that are validated as positive. The next question, any differences on the hits number and immune response if express immunization is applied compared with standard immunization? What's the successful rate of using express immunization, or on what circumstances we would suggest clients to use express immunization? So with actually I can go back to one of the slides, which is right here. So in the bar graph in this particular slide, we directly compared immunization method between standard and express with the same target antigen, and you can see that for standard immunization we got 169 binders, but with express immunization we only got 48. So you can see that with express immunization, at least for this particular target, we did get much fewer binders, but we still got some. And I think we might recommend this coupling of single B-cell screening with express immunization for any client or for any project that are extremely time sensitive. But we can also consider using express or we can start the express and standard immunization at the same time as a backup. So it might be wise to consider that. In case the express immunization doesn't deliver, we can, as a backup, continue on with the standard immunization. So the last question that we'll take now is, if clients need the transgenic mice, do we have the options too? And yes, we do. Currently, we have a CA mouse, and we also have transgenic mice from Harbor and OMT or OmniRat as well, and our corporate BD are working on more collaborations at the moment as well. Well, thank you.